Now, what is happening to the trilaminar germ disc? Now, what else is there besides this trilaminar germ disc? May I show in this? I'll show on the board just for a while. Yes, so this is the trilaminar germ disc that we have. This was the epiblast, this was the hypoblast, this was the primitive streak cells, this was the primitive streak cells, primitive streak, this is the primitive node. From the primitive node, the structure entered in the midline over here and has grown back. This structure is known as notochord. Yes, this is the notochord, and the space beside the notochord, if this is the notochord in the midline, if this is my if this is the is mesodermal area, this is the notochord. So the area, this area beside left side to the notochord and right side to the notochord is the intra-embryonic mesoderm. Yes, this is the intra-embryonic mesoderm. So if I draw the structure once again, this is the ectoderm, this is the endoderm, and here will be the notochord, beside which will be our intra-embryonic mesoderm. So this is the trilaminar germ disc that we have. Not forgetting, here we will have a procaudal plate which is directly attached to the endoderm without there being any underlying mesoderm, and also we'll have an anal plate over here which is also attached to the endoderm without there being any underlying mesoderm. Now what will happen to this trilaminar germ disc? Can we see over here what is happening to this trilaminar germ disc? This trilaminar germ disc will now get folded. Yes, so if this is my trilaminar germ disc, above to my trilaminar germ disc there will be a structure, and below the trilaminar germ disc also will be a structure which is being derived not from the inner cell mass but the trophoblastic cell that I had. Yes, so this is the amniotic cavity, and this is the primary or the secondary yolk sac. This is the secondary yolk sac to be precise. So if this is the yolk sac, this is the amniotic cavity, this is the head end or the cranial end of the embryo, and this is the caudal end of the embryo. Now let us quickly see the development of the gut now. Now, what will happen? There will be folding of embryo. Yes, there will be folding of embryo. What is happening in the folding of embryo? Two foldings that we have. We have a vertical folding, and a horizontal folding. Folding in a horizontal axis and folding in the vertical axis. So it will fold like this, and if this is my embryo, it will fold like this also. So this is the vertical and the horizontal folding. Now what is happening as a consequence of this vertical folding, guys? Yes, if this is the vertical folding of the embryo, this is the head fold of the embryo, this is the tail fold of the embryo. So the procaudal plate that I had here, because of the folding, it has now come over here. Yes, and the anal plate that is there over here, because of the folding, it is now over here. Now this yolk sac which was there, because of the folding of the embryo, it will invaginate inside. And may I use another color? This yolk sac that we have is invaginating inside. Yes, it is invaginating inside and occupying the space like this, forming our primitive gut. This is the primitive gut. Yes, this is the primitive gut, which is attached to this yolk sac, the secondary yolk sac. So this connection between the primitive gut and the yolk sac is known as the vitalo-intestinal duct. Yes, yes. The cranial part is known as the foregut. The middle part, which is being in communication with the yolk sac, is known as the midgut, and the caudal part is known as the hindgut. Can we now see over here? Let me just rub this and draw it very clearly once again a little bit. as I spoke, the foregut, we are having the derivatives such as the floor of the mouth, the tongue, the thyroid, the tracheobronchial tree, the esophagus, and the stomach. Then we have the stomach and the first part of the duodenum, and the second part of the duodenum, only the upper part till the insertion of the ampulla of Witter. Then we have then we have the midgut. What are the derivatives of the midgut? From the second part of the duodenum, third part of the duodenum, the jejunum, ileum, and the appendix, and also the cecum, the ascending colon, the right half of the transverse colon. And also the derivatives of the midgut are the liver, the pancreas. Yes, and then we have the derivatives of the hindgut, which include from the left half of the transverse colon, the descending colon, sigmoid colon, and the rectum, and only the upper part of the anal canal, not the lower part. Where is the lower part of the anal canal being derived from? The upper part of the anal canal is being derived from the endoderm, and the lower part, the lower part of the anal canal is being derived from the ectoderm, and it also has a different innervation. Yes, other thing. So this is the entire gut tube that we have formed. The esophagus, to start with the esophagus is a muscular tube that is extending from the pharynx till the stomach. Yes, that is the tube. So the part of the foregut that I spoke over here just now on the board, the foregut, from the pharynx till the stomach, the part of the foregut is forming our esophagus. Firstly, the esophagus was not canalized. It was obliterated. The esophagus will be obliterated, and after that there will be recanalization of the esophagus. And one more thing that is happening over here is from this tube only there will be a diverticulum which is arising. Yes, that diverticulum is known as laryngotracheal diverticulum. That laryngotracheal diverticulum is forming a tracheobronchial tree. This laryngotracheal diverticulum is being separated from the esophagus by a septum known as the tracheoesophageal septum. So there are some clinical abnormalities which sir will talk about if this septum is not being separated. Yes, then what happens? This is forming only the mucosa and submucosa part of the esophagus. The foregut is forming only the mucosa and submucosa part of the esophagus. From where is the muscularis mucosa, serosa, and all connective tissue, blood vessels, glands, and all are derived from? They are derived from the splanchnopluric layer of the intraembryonic mesoderm. Just I'll say one point over here. Yes, so just I'll say one point over here that I spoke. So I'll have to carry this, I think. Yes. So if this is the ectoderm, this is the endoderm, and this is the mesoderm, this is the notochord. So where we are seeing the embryo from, if this is the human being, we are seeing from here. So this is the transverse section. We are cutting from here and seeing from here. So this is the notochord in the midline. This is the notochord in the midline. This is the notochord. So above is the ectoderm, below is the endoderm, and here in between here entire thing will be our mesoderm. So this mesoderm is the intraembryonic mesoderm that we have. So it will have a split like this. It will have a split. So the upper part of the intraembryonic mesoderm is known as the somatopluric layer of the intraembryonic mesoderm. This is the splanchnopluric, and here we'll have the invagination of the yolk sac and all, everything forming the foregut, midgut, and hindgut. So that is the thing. So can you see this splanchnopluric layer of the intraembryonic mesoderm is surrounding the foregut. So this splanchnopluric layer will form what layers? The muscularis mucosa, the serosa, and the muscularis externa part of our gut wall. So I think this is all I have to talk about the development of esophagus. I hope it was understandable. Thank you, sir, once again.